I'm going to make a start and move on to the next talk, which is hosted by Dave Madge, Vice President of Research Service Division at WuXi AppTec.
Good morning, everybody. Thanks a lot for being here. It's great to be back at an in-person event. So I'm based in Cambridge, and I support the preclinical services activities for WuXi AppTec. What I'm going to talk about this morning is our platform for target discovery, validation, and further development in the immuno-oncology space, hopefully over about 30 minutes or so, and not keeping you from your coffee break. So let me just make sure everything works here. Okay. So firstly, a very brief introduction to WuXi. Hopefully many of you are familiar with the company at a high level. We're a global company. We were founded about 20 years ago. Many sites in China, the US, and more recently adding sites in Germany, in the UK, Switzerland, Israel, Korea, and Japan. Supporting all activities across the drug discovery space. We're not sensitive to modality or therapeutic area, and we'll come back to that as a topic as we go through some of these slides. Today, we work with over 5,000 different collaborators and clients. So these, as you can tell from some of the names on this slide, are very diverse. We work a lot with small companies, academic teams, spin-out groups from universities, right up through, as you would imagine, to multinational corporations. But our philosophy throughout this activity is to build an end-to-end platform so that we can support drug discovery programs of diverse kinds from start to finish, from target selection, from initial idea, right through to commercial realization, right through to patients. And we will do that across different modalities and different therapeutic areas. I mentioned some of the major areas of our activity at the bottom of this slide. I'm going to focus almost entirely at the left-hand side of this slide this morning, but be very happy to engage with you offline on any of the other topics mentioned here. So it's expanding a little bit on that philosophy about building an end-to-end platform. As you can imagine, we're in the small molecule drug discovery space. That entails many, many different activities. And what we really want to be able to do is to support you across the whole range of these activities, not necessarily with every one of these kind of assays or services, but to give you the flexibility to come and cherry pick amongst these according to what your needs are and how they support your own particular activities. So a little bit more detail here from target selection through to traditional medicinal chemistry, optimization, in vitro and in vivo biology, right through to delivering a preclinical candidate, which is really the space that my team are focused upon. So turning in a little bit more detail to the immuno-oncology space, there's some data on this slide that comes from a review published in early 2020, and it really compares the pipeline with respect to targets and modalities over the preceding 2 years. And essentially what you see is, as we all know, I think, massive growth in the number of different targets being addressed in the immuno-oncology space, almost a twofold increase in the number of targets that are at some stage or other in preclinical or early clinical development. But also a massive increase in the diversity of modalities that are being addressed here. So not only small molecule therapeutics, of course, also oligonucleotides, all kinds of vaccine therapies, oncolytic viruses, and more diverse modalities, all of which need to be supported through this pipeline of activity of discovery, validation, optimization, through to clinical candidate and beyond. So this really is where we come in, and over the next 15 minutes or so, I'm going to drill down into some detail across these major buckets of our activity through in vitro screening for target ID and validation, particularly using genetic assays, through in vitro immunology assays, a little bit about syngenetic and humanized models, and then a little bit more about immune profiling and biomarker discovery. So as you can imagine, this is a platform that supports programs right through to clinical development. With respect to target discovery and validation, some of the main areas of our activity shown on this slide. Of course, phenotypic and functional studies, Major emphasis on gene manipulation as a tool for supporting target ID, and I'll talk mostly over the next couple of slides about CRISPR-related tools, but also a large cell bank, around about 1,000 cell lines, most of which are genotyped, to allow us to go rapidly into relevant cell lines and really understand whether the target that we've identified, perhaps through a CRISPR screen, is really playing the role that we're expecting it to play once we go into a cell line. And then following that up with in vivo models using Madge genetics and in vivo gene editing. So the major tools here for the genomic manipulation platform, raw cell line manipulation in generic cell lines, typically using KO, KI knockdowns, sgRNA library screens, and I'll show some data for synthetic lethality screens and related screens in a few slides, but also taking those tools into primary immune cells and also being able, therefore, to manipulate primary immune cells and have a model that's a little bit closer to real life. And then phenotypic assays and selective knockdown using tools other than genetic manipulation, such as PROTAC, DTAG, SMASH, and other molecular tools. So an overview of CRISPR libraries and screens. I suspect many of you are very familiar with this kind of breakdown. So using knockout libraries, looking at repression and looking at activation, so we can run these assays in a couple of different modes. Synthetic lethal screens, typically looking for dropout and identifying a list of essential genes. We can also run this in a negative mode, a negative screen, that would identify genes that have become essential under applied selection conditions, and also under a positive screen, looking at identifying genes that convey a survival advantage. And I'll show a few examples of this kind of thing in a couple of slides' time. But this is the sort of a high-level overview of the workflow and identifying some of the tools that we're using on a regular basis. So short hairpin RNA libraries, sgRNA libraries, manipulating these through signaling pathways, through disease association genes, and of course looking in some cases at whole genome screens. Running these as synthetic lethality and resistance or selectivity screens. And then of course using NGS to identify exactly what's going on. A couple of examples here on what that looks like in practice. So top right of this slide, sensitivity assay gene target identified here. So what we're seeing is a rightward shift from the dose response to a test article from a naive cell line in the red to a sensitized, to a desensitized cell line in the right, where we've knocked down a gene that confers drug resistance. And then the lower panel, a leftward shift in the similar kind of assay where we've knocked out a gene related to drug sensitivity. So you can see how quickly we can build assays that allow us to confirm and validate targets, and indeed look at the sensitivity of those knockouts or indeed knock-ins to different kinds of test articles. We can also do this with double knockout, so running many both single and double knockout screens in cancer cells. And the slide shows a few examples of the kinds of experiments that we can do. So cell lines with single or double target knockout, generating using CRISPR, Cas9, and then taking these, validating these, and then taking these into highly bespoke assays. So we're not only using sort of more traditional double-strand break editing, we're also using the more recent Nick-style editing, CRISPR prime editing, to give us a more precise editing tool and a more reliable result with less off-target liability. So this has become a standard way of ensuring the quality of the data that we generate. These technologies, as I mentioned earlier, are not restricted to naive cells. We can take these technologies into immune cells of various types. And I show some data here exemplifying taking these technologies into mouse T cells, human T cells, monocytes, dendritic cells, right through to human M1 and M2 cells. And we see very good alignment between using the gene editing to using whether we record it with sequencing or whether we record it using fax-based screening. So this gives us a way to move forward into a more complex model system using an increasingly relevant cell line to confirm validation, and to start to create models that we can use to really understand how we're optimizing molecules towards these targets. To back that up with real samples, real cell lines, we have a panel of around 1,000 cancer cell lines that we can run in high throughput mode with a variety of different endpoints. These are very diverse. They represent all the major classes of human oncology. They're also geographically diverse, so we're sourcing from global resources. As I mentioned on the right-hand side of this slide, we can run these in a high throughput mode to support screening. We can run them with many different endpoints, including protein expression profiling. And we can run them in more complex 3D assays as well, so we can really get a good understanding for the sensitivity of a specific cell line with a specific mutation to molecules that are manipulating that molecule, that gene or that pathway. So having carried out that step, having identified targets and validated targets, we might now want to identify small molecules that are a good way to manipulate those targets and to move towards a therapeutic. So this slide provides a high-level overview of our lead generation platforms, starting with high throughput screening, which could be biochemical, could be phenotypic, could be high content screening. Moving down through affinity-based selection methods for small molecules and peptides and oligos, which could be ASMS, it could be using DNA-encoded libraries. So we have a very large panel of DNA-encoded libraries, around 80 billion small molecules tagged with DNA that we can run very, very quickly through a hit-finding platform by affinity. Fragment-based screening with all of the supporting structural biology so that we can rapidly map out binding sites of fragments and then follow up with an understanding of where each of those fragments binds using crystallography. And then, of course, virtual screening. So our computational platform allows us to rapidly screen large libraries, billions of virtual small molecules to identify candidates for synthesis and follow up with screening. So these platforms are all enabled across multiple sites in our organization and integrated with our follow-on medicinal chemistry and in vitro biology. Some of that downstream in vitro biology might be cherry-picked from this extensive list. So we're agnostic with respect to target class, working across all kinds of the targets that you might identify. And also very flexible with respect to the endpoints of each of the assays that we might build across all these different target classes and assay formats. All of these assays being available in high throughput mode, so most of our sites are enabled with 384 and 1536 well plate screening facilities. And of course, we have compound collections that we can apply to these assays. By and large, though, these being used to support lead optimization in a medium throughput mode and highly integrated with medicinal chemistry and DMPK. So turning briefly to how we might follow up with molecules that we've identified that are modulating targets that we've identified, looking to the slightly more complex scenario of understanding immune cells in the tumor microenvironment. These are the different ways in which we might, different cell types that we might want to examine. We might want to understand how our molecules are impacting on what's happening in these different cell types. I'll briefly, over the next few minutes, show some assays for a couple of different scenarios. In this setting, will be by no means comprehensive. So if there are areas here that you'd like more details on, please feel free to contact me or one of my colleagues. But of course, we do a lot of work with mixed lymphocyte systems, looking in detail at the synapse, so the immune synapse. And this, of course, is in particular where we might be looking to understand the efficacy of an immune checkpoint inhibitor. So looking at the synapse between the dendritic cell and the T cell, we can imagine many different kinds of endpoints that we could apply in this setting. I provide a little bit of data here, which looks at, in the top panel, the activation state, and in the bottom panel, the responses to a well-known antibody, anti-PD-1 antibodies, with respect to cytokine production, so interferon gamma, interleukin-2, in the first and second panels in the bottom. But of course, we can also look at this in conjunction with other modalities. So the bottom right panel is looking at the efficacy of a small molecule ligand for one of the synapse proteins alongside an anti-PD-1 antibody. And what you see, of course, is a much more pronounced effect. In this case, we're measuring interferon gamma as an endpoint. So this is a good way for us to rapidly understand the benefits of co-mixing or the synergy between different modalities and different pathway manipulations in a mixed lymphocyte assay. We also spend a lot of time looking at differentiation of helper T cells. The pathway I'm sure you'll all be familiar with on the left-hand side of this panel. The endpoints will typically being used to identify whether a particular manipulation of a particular target is driving a naive T cell towards Th1, Th2, Th17, or Treg, as we show on this slide, and in the main using cytokines like interferon gamma, IL-4, IL-17 measurements to really understand which phenotype is being driven by manipulation with different parts of this pathway. Looking at macrophage polarization is also a fairly common way of understanding how our molecule might be manipulating what's happening in the T cell and the tumor microenvironment, and in particular looking at the ratio of M1 to the various M2 subclasses of macrophage. So this is an assay that's set up in our organization, in particular looking at cytokine endpoints, IL-6, TNF-alpha, and IL-10 shown on this panel, and really using those cytokine endpoints to understand the ratio of macrophages that's being triggered by a particular intervention. Similarly, for macrophages, phagocytosis assays are something we're very frequently turning to, and on this slide we're representing a measurement of increased phagocytosis in response to anti-CD4 and anti-CD47 in the middle panels. But we can also run myeloid-derived suppressor cell assays in a similar kind of mode and really understand how we're impacting on phagocytosis with different kinds of intervention. Similarly with natural killer cells, I won't go through this in detail, but the endpoints are in particular enabled by having access to different kinds of imaging and cell sorting technologies, and we show data at the top of here where we're sorting by both NK and RAGI, so that gives us a very quick indication of, for example, the ET ratio with different treatment paradigms. All of that is perhaps a little bit abstract, but to bring this sort of closer to a more complex system, a more integrated system, we're very frequently working now in systems that are derived or built around multiple cell components, each being manipulated in different ways, and understanding what happens when we build these more complex systems and what happens when we intervene at different stages or with different elements of these processes is critical to understanding how these assays and how the molecules manipulating these targets will translate into in vivo assays and ultimately into man. I'll show an example here where we've built an assay around the NLRP3 inflammasome system. So we're using PMA differentiated THP1 cells, stimulating with LPS, and then following treatment with nigericin, the toxic microbial product, to further stimulate, and then looking at inhibition of the cytokine production, IL-1 beta, in this case in the middle panel, and also the endpoint of caspase activity in the top right panel in response to increasing concentrations of a well-known modulator of this pathway, MCC 950. So not only can we produce these complex assays and measure them and really get useful endpoints out of them, we're also producing all of the proteins involved in building these assays, so we can very quickly make genetic changes to specific proteins and create highly specified versions of these kind of assays. I've exemplified this with NLRP3, but as you can imagine, there are many other areas we have these kind of more complex systems. One that springs to mind is the STING pathway, where again we've created a highly integrated assay system. So those are a fairly comprehensive collection of in vitro assays. Of course, we also want to be able to demonstrate in vivo activity, show a high-level overview here of some of our syngenetic models. These are all genetically characterized. They're run typically with endpoints like tumor volume. Currently running at any one time maybe 50 or 60 of these assays in these mouse models. Not all modalities are appropriate for using those kind of assays without some additional manipulation, however. So in particular, if we're working with humanized antibodies, we might use human PBMC adoptive transfer to create a mouse model that's much more tailored to the human antibody. We show a little bit of data in the top left-hand panel where we've done that, and build mouse models around that to give us something that's much more bespoke for human antibody. Similarly, on the right-hand side, we can also use human target gene knock-in to create models in this space. So that gives us a way of ensuring that we're looking at modulation of the human target in these animal models. We also work quite a lot in bispecific T-cell engagers, and again, need to create very often bespoke models that are relevant for this modality. In this case, we're using, again, HPMC adoptive transfer to create a model that's then sensitive to a bispecific T-cell engager. All of these things can be followed through into clinical development. So we've built a CAP-certified and GCP-compliant lab system for clinical biomarkers and pathology services. So as we're running through, in particular, the later stages of an oncology discovery program, we'll be thinking about how we might support that in the clinic with diagnostic tools and biomarker tools, and making sure that we're able to do that and engaging with the clinical teams so that we can support these endpoints during the clinical development process. So I will stop there and be happy to take any questions, but thank you very much for listening today, and thank you for being here.
Caught me off guard. Okay, so
Early
let's see
coffee
if
break.
Okay, so we've had one question come through during the presentation. We are about 7 minutes early, so if you do have any additional questions that you would like to submit, please go through onto Swapcard, onto the Q&A for the auditorium, and submit any comments or any questions that you would like to put forward. So the question is, what will you do if you identify known targets such as HER2 or PD-L1?
Mm-hmm. Yeah, of course, many target identification screens will identify targets that people are already working on. We are just to be very clear, we're a service provider. We're working with our clients to help them identify targets and validate targets. If somebody wants to look at new modalities for existing targets, or feels the need to identify new molecules for HER2 or PD-L1, be it antibody, small molecule, or genetic tool, then of course, we're perfectly able to support multiple clients working on the same target. We are not generating our own intellectual property. We're generating data on behalf of our clients.
Okay, fantastic. If no one's, I can't see anyone. The thing is, I can see people typing, but I'm not sure if there's something. Someone saved me. Thank you. In the PBMC adoptive transfer, do you see GVHD? When does it kick in? And do you offer the B2M mice to avoid GVHD?
So I can answer the last part of the question. We do offer the B2M mice to avoid GVHD. Off the top of my head, I don't know when GVHD kicks in, so I would need to discuss that with some colleagues, and I assume I will get a list of who's asking the questions and can do that.
It's anonymous,
directly.
So unfortunately Anonymous would have to come and find you. So if you have just submitted that question, please head over to Whooshy's booth to continue that conversation and find out the answer to that question. Is anyone else typing any questions? If not, I'm happy for someone to put their hand up and ask a question if they prefer not to use Slido, but if not, I don't want to leave Dave here on his own. So if I don't see any questions, I'm going to end the session there. Okay, I'm going to end the session there. Thank you so much for your presentation. Okay, so that does bring a close to